Vaccines are one of the most effective tools in modern medicine, saving millions of lives each year by preventing infectious diseases. The journey of vaccine development is a meticulous process that involves extensive research, rigorous testing, and careful monitoring. This article will delve into the detailed stages of vaccine development, from the initial discovery of a pathogen to the distribution of a safe and effective vaccine to the public. 1. Understanding the pathogen. Identifying the pathogen. The first step in vaccine development is to identify the pathogen that causes the disease. Pathogens can be viruses, bacteria, fungi, or parasites. Scientists study the pathogen's structure, genetic makeup, and life cycle to understand how it infects humans and causes disease. Studying the immune response. Understanding how the human immune system responds to the pathogen is crucial. Researchers investigate which parts of the pathogen trigger an immune response and how the body defends itself against the infection. This knowledge helps in designing a vaccine that can mimic the natural immune response without causing disease. 2. Vaccine design and development. Selecting the vaccine type. There are several types of vaccines, each with its own method of inducing immunity. Live attenuated vaccines use a weakened form of the pathogen that cannot cause disease. Inactivated vaccines use a killed version of the pathogen. Subunit, recombinant, and conjugate vaccines use pieces of the pathogen, such as proteins or sugars. mRNA and viral vector vaccines use genetic material to instruct cells to produce an immune response. Laboratory research. Once the vaccine type is selected, scientists conduct laboratory research to develop a prototype vaccine. This involves creating and testing the vaccine in cell cultures and animal models to evaluate its safety and efficacy. 3. Preclinical testing. Animal studies. Before testing in humans, the prototype vaccine undergoes extensive testing in animals. Researchers assess the vaccine's ability to induce an immune response and its safety profile. This stage is critical for identifying any potential issues and refining the vaccine formulation. Toxicology studies. Toxicology studies are conducted to ensure that the vaccine does not cause harmful side effects. These studies evaluate the effects of the vaccine on different organ systems and determine safe dosage levels. 4. Clinical trials. Phase 1 trials. Phase 1 trials involve a small group of healthy volunteers and focus on evaluating the vaccine's safety, dosage, and ability to induce an immune response. These trials help identify the appropriate dose and any immediate side effects. Phase 2 trials. Phase 2 trials involve a larger group of participants and further assess the vaccine's safety and immunogenicity. These trials provide more data on the immune response and help refine the vaccine formulation. Phase 3 trials. Phase 3 trials are large-scale studies involving thousands of participants. These trials are designed to evaluate the vaccine's efficacy in preventing disease and monitor for rare side effects. Phase 3 trials provide the data needed for regulatory approval. 5. Regulatory approval. Submission to regulatory agencies. After successful Phase 3 trials, the vaccine manufacturer submits a comprehensive dossier to regulatory agencies, such as the FDA, Food and Drug Administration, or EMA, European Medicines Agency. This dossier includes data from preclinical and clinical studies, as well as information on the vaccine's manufacturing process. Review and approval. Regulatory agencies review the submitted data to ensure the vaccine meets stringent safety, efficacy, and quality standards. This process may involve independent expert committees and public hearings. If the vaccine meets all requirements, it receives regulatory approval for use. 6. Manufacturing and distribution. Scaling up production. Once approved, the vaccine must be produced on a large scale. This involves establishing manufacturing facilities that meet strict quality control standards. The production process must be consistent to ensure each batch of the vaccine is safe and effective. Quality control. Quality control is a critical component of vaccine manufacturing. Each batch undergoes rigorous testing to ensure it meets safety and efficacy standards. This includes testing for contaminants, potency, and stability. Distribution and logistics. After manufacturing, the vaccine is distributed to healthcare providers and vaccination centers. Effective distribution requires careful planning to ensure the vaccine remains stable and potent during transportation and storage. Cold chain logistics are often necessary to maintain the required temperature conditions. 7. Post-market surveillance. Monitoring safety and efficacy. Even after a vaccine is approved and distributed, its safety and efficacy continue to be monitored. This is done through post-market surveillance systems that track any adverse events and evaluate long-term effectiveness. Health agencies may conduct additional studies and collect data from healthcare providers and patients. Addressing adverse events. If any safety concerns arise, regulatory agencies and manufacturers investigate the issues and take appropriate actions. This may include updating vaccine labels, issuing safety warnings, or in rare cases, recalling the vaccine. 8. Public health impact. Achieving herd immunity. Vaccination not only protects individuals but also contributes to herd immunity, reducing the spread of infectious diseases within the community. High vaccination coverage is essential for controlling and eventually eradicating diseases. Global vaccination efforts. Global vaccination efforts, often led by organizations like the World Health Organization, WHO, and UNICEF, aim to ensure equitable access to vaccines, especially in low and middle-income countries. These efforts are critical for controlling outbreaks and protecting vulnerable populations. Conclusion. The development of a vaccine is a complex and multi-step process that requires collaboration among scientists, regulatory agencies, healthcare providers, and public health organizations. From understanding the pathogen to post-market surveillance, each stage is crucial to ensuring the safety and efficacy of the vaccine. The ultimate goal is to protect individuals and communities from infectious diseases and improve global health. Thanks. Please like, share, and subscribe.